So last year, this time, like September-ish, Shire, we were shopping for a new platform for immunoassays. And we did our homework and due diligence and everything, and then we compared 3 other platforms, and we acquired SIMOA HD1 Analyzer. So today I'm going to talk my experiences using this new instrument for me, and also talk about challenges that we faced, and also how we overcome them. So that's why the long and winding road for RANKL assay. So before that, I also wanted to say that at Shire, I'm in bioanalytical and biomarker development department, and we support discovery through clinical trials, the whole with the assays that come with, and we develop them and validate them or send it out to a CRO for validations. Before going into the assay development, I wanted to introduce RANKL to you if you guys are not familiar with RANKL. RANKL is a receptor activator of NF-kappa B ligand, and RANKL is a part of TNF superfamily, I should say, more than alpha. So you can, RANKL can be seen in 3 isoforms. First 2 are basically the splice variants. They have transmembrane domain, you can see here, dark. And the 3rd isoform, this transmembrane domain from these 2 isoforms will be truncated, and what you see here is a soluble form. And this is the predominant form that you see in serum or plasma. And this soluble form of RANKL was earlier used to evaluate various pathologies, as you can see, a wide variety of them: osteoporosis, also periodontal, immunological disorders, arthritis, oncology, to name a few. So here I'm going to show you one of the pathways RANKL is involved in, and specifically for bone strength. All of us who are sitting here, and everyone, even other animals, you have these cells called osteoblasts, and these are nothing but differentiated from mesenchymal stem cells. And these, when they undergo stimulation from parathyroid hormone or PGE2 or even vitamin D, they differentiate into bone-forming cells. And a mature bone have these cells called osteocytes. And these osteocytes produce RANKL. Not just osteocytes, other immune cells also produce RANKL. And the RANKL activates another type of cell called osteoclasts. These are multinucleated cells. And they undergo bone resorption. There's another protein called OPG. OPG binds, sequesters RANKL from activating osteoclasts. So this RANKL, just by itself, binds RANK receptor and activates osteoclasts. So OPG and RANKL involves in homeostasis for this bone formation and bone resorption. And the soluble form of RANKL, the RANKL that we're looking at, is an investigational serum protein biomarker in bone strength and bone resorption. So RANKL concentration is inversely proportional to bone mass. So higher concentration of RANKL is involved in lower bone mass. Switching gears now and talk about immunoassay development. There are quite a few commercial kits that are available out there. And here I'm highlighting 3 of them. Biomedica, a German company, they have a dynamic range for RANKL 3.75 to 125 picograms per ml, and they use recombinant RANKL as a calibrator. Here what you can notice is it measures soluble free RANKL, and there's another company, a Czech company, BioVendor. It has a different range, and it's serum-based RANKL calibrator they use, and it measures total. As you can see, the values that you're getting here in healthy volunteers is quite different. And even if you look at just free soluble, humans have a wide range of RANKL in their serum. And another kit that I'm talking about here is R&D Biosystems. And they have a different range. They use recombinant as well, and they don't talk much about what kind of RANKL that they're going to measure. What I want to point out here is the amount of RANKL that you see in sera totally depends on the kit that you use and what you are measuring, and you don't know exactly. And they are not, I'm not saying that they are not sensitive, but they're not sensitive enough to measure RANKL in all human sera, because as you can see, the errors are pretty high. So the goal of my project is to develop a reliable and highly sensitive assay and detect RANKL in human sera that outperforms the commercial assays. For that, now we have a new tool, SIMOA Technology, and they say it's reliable, very ultra-sensitive, so we want to try this out. And thank you to the earlier presenters. We got a lot of background about SIMOA. So I'm going to skip this slide and go into the next step in assay development. One of the major criteria, even Ralph mentioned this earlier, is to pick an antibody pair. We need an antibody pair because one is a capture antibody similar to any immunoassay, another one is a detector antibody. So we went ahead and picked R&D Systems DioSet antibody pair because we can get larger quantities of the antibody. Also, there's a lot of literature available about these antibodies for RANKL assay. And also, R&D Systems by itself is widely used in various, both in universities and industry laboratories, so why don't I go ahead and use that. So here is an example of a regular conventional ELISA, where you can see OD on y-axis and concentration of RANKL. It's a recombinant RANKL that they supplied with the DioSet. And these are the capture antibodies and detector antibodies. And when you look at it, these should pick all three isoforms of RANKL, the way they immunize the animals to generate antibodies. What we are getting is limit of detection of 78 picograms per ml in regular ELISA. One point to note is to start with the OD that I'm getting is a little high. That is higher for an ELISA. We thought it was a concern earlier, but I thought it's good. Let me try it on SIMOA and see how they perform. So for that, we got homebrew kits from Quantification and made our own bead conjugates and biotin-related antibody. And all these are ready to use, supplied in the kits. When we put them in SIMOA, this is the first SIMOA data that we got for RANKL. We used two different concentrations of RANKL, recombinant RANKL, and no RANKL background control. And the readouts of SIMOA is all in AEB, that is average enzyme per bead. What you can see is with just no RANKL negative control, the background is so high. That was surprising for me. And also in the two concentrations of RANKL that we used, the signal-to-noise ratio is not good at all. So there's no dose-dependent increase when we use recombinant RANKL. Usually, when you have an antibody pair that works in ELISA, it typically should work in other immunoassay platforms as well. Even Ralph was mentioning about this in his talk. It's a new platform, so we were kind of worried, like, okay, where is this going? But we spoke with Quantification, always in touch with all the scientists, most of the scientists there, and we did troubleshoot quite a few things, reversing antibodies, like the capture, we made it into detector and detector to capture, concentrations, and what all parameters we could test. But what we came out is that dual set antibodies for RANKL, we couldn't transfer into a SIMOA platform. And we went back to the drawing board and looked for various antibodies that are out there, and we picked 12 commercial antibodies. Another thing I want to point out is in pharmaceutical companies, you don't have ample amount of time to search everything and available, so you have a specific amount of time, and we want to make sure that we have an assay that is working for us. So we tested 12 commercial antibodies for RANKL, both in ELISA and also in SIMOA platforms. And the criteria for selection is low background and dose response. Then we finalized with the capture antibody that is mouse monoclonal and detector antibody that is goat polyclonal, two from different vendors. And interestingly, they both are immunized by a similar RANKL immunogen. And this pair should also detect all isoforms of RANKL. When we tested in ELISA, an approximate sensitivity is 5 picograms per ml. It is different from, if you remember, for Dioset, it was 78 picograms per ml. Voila, I mean, we tried this in SIMOA. You have AEB on y-axis and nanograms per ml RANKL. You can see a limit of detection of approximately 100 femtograms per ml, way better compared to the Dioset and also ELISA, because it is 24 more sensitive than the ELISA I showed you earlier that was 5 picograms per ml. But again, this is the first time we ran on SIMOA, and the background was 0.04 AUB. When we spoke with the quantitative scientists, they say typically you should see anything below 0.02 AUB should be a background. So we wanted to optimize this assay. So we went ahead and did some additional assay optimizations. We selected various parameters that we can test or try, and we also went with homogeneous or sequential, where in homogeneous you mix your detector, capture antibody, sample all together, or sequential, which is one after another. I also looked at calibration curves and fitting and weighting. We have other assays that we are developing right now on SIMOA, and we are using DOE for that to optimize these conditions to come up with a good calibration curve. And here is an optimized RANKL quantification on SIMOA. Again, AEBs on Y and concentration of RANKL. And the lower concentrations are so close to x-axis, so I just put an insert here showing that its curve fitting is pretty good. And limit of detection, we improved it to 50 femtograms per ml, better than the previous data that I showed. And the background has come down, but it is not as good as what we wanted, but still we can live with it because signal to noise ratio is good enough. Dynamic range, dynamic range 100 femtograms per ml. I wanted to bring it to attention that its limit of detection is different from dynamic range, and I'm going to show more data about that. CVs look good, 4PL it is, and calibrators is the same calibrator that I'm using from R&D systems. It's a recombinant RANKL. So we have an assay where the quantification looks good. It looks very sensitive. Next step is to qualify or validate this assay so we can use for either non-clinical or clinical studies. Here I wanted to mention that in the field of biomarker, there's a paper published, a white paper published in 2006 by Lee et al. We still use this paper because FDA doesn't have guidance for the biomarker validations. So this paper explains about all the steps that we have to use for validating an assay. As I mentioned earlier at Shire, we develop an assay and pre-qualify them and send it out to a CRO for full validation. And we picked Frontage Laboratories as our CROs to validate this assay, and right now we are in the process of assay transfer. Where for pre-qualifications, we tested MRD, that is for matrix effect, as Ralph was talking about, assay precision, parallelism. I'll go into more details about some of these. So I'm going to mostly talk about the top 4 because of time limits. Here is minimum required dilution. The first thing we always do is to test how much matrix effect you are seeing. because we are going to use this assay for both clinical and non-clinical in serum samples or plasma, whichever one you want to check. But you want to make sure that there is no effect when there's something in the serum that is affecting your assay. So for that, here I'm showing you 4 different dilutions of serum. This is a depleted, we made some depleted normal human serum, pooled normal human serum, and tried 4 different dilutions. And what we can see here is 1 to 15 dilution, that is roughly 6.67, has not a big difference or acceptable difference compared to the buffer. So from here onwards, my experiments will be all done in 1 to 15 dilution of the serum. Next step is spike recoveries. Is nothing but precision. And since it's pre-qualifications, we didn't want to do 5 replicates. We just did 3. And another small hurdle here, what you can see is I have three levels of calibrators, 100, that is closer to the upper level of the curve, 10 picograms per ml in between, and 0.5. All the time, so many times we repeated, in the 0.5, the bottom of the curve, the spike recoveries were failing. So we studied more, we looked at the curves more, and what we have identified is that the concentrations are coming so close to x-axis, very ultra-sensitive here. What's happening is that the curve fitting might have a problem when you're coming to lower part of the curve. What I show you here is SIMOA data, where there are 2 curves. One is the red one. What you have is the one that I was using for this slide, where I have picograms per ml, only 6 levels. And I wanted to show you the insight to make it easier. And also, what's happening is curve fit is here, but what we realized is any gaps in here is kind of creating problem for precision studies. So we included more points at the bottom of the curve, and they are fitting differently compared to this. And this minor change is affecting my spike recoveries. It's because we are going into that ultra-sensitive mode where you wanted to be very careful to get your LLOQ. So from here onwards, I was using 9 points in the curve so that my precision should be good. And I tried that. Here the same, repeating the precision data, 3 different assays and 5 different levels of RANKL calibrator. And you can see the mean pretty much very good, and this percent CV is good. And one thing I wanted to point out is the lower one. I mean, we were able to stretch it to 100 picograms per ml dynamic range, but the percent CV is 25.4. In the field of biomarker, you know about fit for purpose. So can you say 25.4, is it good enough percent CV? So it depends on your studies that you plan. You can include the concentration of 0.1, or you have to go a little above for your dynamic range. The next important thing, again, going back to Ralph's study, talking about the spikes that we are doing at the calibrator is a recombinant RANKL, but what we are measuring in serum is endogenous. There's a huge difference between recombinant and endogenous. So the paper that I was showing you earlier, they talk about parallelism. What you do is you pick three samples or 3 healthy or any human sera that has high RANKL, endogenous RANKL, and you dilute them, serially dilute them. Here I serially diluted 1 to 5-fold, and see if they are parallel with your buffer or your reference standard. If it is parallel, that shows that your dilution linearity is working well. That means your assay will perform well even when you use endogenous, when you're looking at endogenous RANKL. Here there is, things are a little rough here because it's above MRD. I was using 1 to 15 dilution as MRD. Anything above MRD, there is a bumpy signal here. And also looked at specificity, where you have a binding partner, OPG. I was talking about that sequesters RANKL. So here I have two curves. This is a regular conventional ELISA, where you have the blue curve is with OPG, and the red curve is without OPG, and there is no difference between both. That shows that OPG, a binding partner, won't influence the assay, and also the assay can pick both bound RANKL and free RANKL. It's all good. I mean, we have a pre-qualified assay that's working good in human serum. Next step comes to actual comparison, how good it is with ELISA when it comes to real samples. So here we studied 45 healthy human sera and 15 disease sera. Out of 15, we have both osteoporosis and RA patient sera here. And you have a lot of data here. What I wanted to show you is on the left two, these two are normal and diseased RANKL concentrations in picograms per ml on SIMOA. And here you have is ELISA kit, that DioCert ELISA kit that I was testing earlier. And the blue bar shows the LLOQ of SIMOA, what we are getting, and the red bar is LLOQ of ELISA that R&D systems described in the kit. Even though, I mean, first point is you can see a significant difference between normal and disease in RANKL concentrations, both in SIMOA and ELISA. But one thing you have to notice is that clearly, even though you're detecting RANKL concentrations in ELISA, all are below quantifiable range. So whatever data we get here, we cannot say it is quantifiable. So, but when it comes to SIMOA, we have more than 85 or 90% of the samples what we tested are in quantifiable range. But in ELISA, we got only like 33% that were in quantifiable range. There's a huge difference between SIMOA and conventional ELISA. So one more point I wanted to bring it up is it's hard to compare. When you say correlation studies, we can do statistics, but it's hard to compare because the antibody pairs we are using for ELISA and SIMOA are different. So, but still I did a correlation studies, and the slope of the curve is 0.98, is pretty good. And also correlation coefficient is 0.51. So it's a good comparison, and we feel like SIMOA is a way to go for the assay for any studies that we are doing here onwards for RANKL. Summary. So the SIMOA assay that we developed for RANKL has a 5-log dynamic range, and LOD of 50 and LLOQ of 100 femtograms per ml. And the sensitivity was improved by adding calibrated levels at the bottom of the curve. We demonstrated acceptable sensitivity, precision, and also parallelism. And 85% of the samples analyzed in SIMOA platform are quantifiable range. In comparison, in ELISA, only 32% of the samples. And one important point I want to make is analytical platform in combination with the choice of antibody pair, that dictates the sensitivity and the performance of the assay, not just the antibody pair. So we also need the platform. So again, this is Shire group that, so here is our department head and my supervisor, a colleague, another person. Tom is he's working on other assays, other SIMOA assays, and people who helped me at Quanterix during my troubleshooting steps. And thank you, and ready for any questions. Yeah.
2 words, helper beads. Have you thought about using helper beads to increase the precision at the low end of the assay?
No, I did not use it. Maybe I should talk to you after.
So these are non-functional beads that you can lower the number of functional beads that you would use, replace them with these non-functional beads, and it limits your top end, but it can also add precision in the low end. We can talk about that afterwards.
Yes, thank you.